Methods in field biology. Methods of estimating population density of animals and plants. ChatGPT said. Methods in field biology. Estimating population density of animals and plants. Introduction. Estimating population density is a fundamental aspect of field biology, essential for understanding ecological interactions, conservation status, and population dynamics of species. Population density refers to the number of individuals per unit area or volume. Various methods are employed depending on the species' mobility, habitat, and behavior. These methods can be broadly classified into direct and indirect methods. The choice of method depends on the accuracy required, the ecological characteristics of the species, and the feasibility of data collection. Methods for estimating population density of animals. 1. Direct methods. These involve counting individuals within a specific area and are generally used for organisms that are relatively immobile or easily observable. A quadrat sampling for sessile or slow-moving animals. Quadrat sampling involves placing a defined sampling unit quadrat within a study area and counting the individuals within it. The density is calculated as density equals total individuals counted in quadrats total area of quadrats density equals total area of quadrats total individuals counted in quadrats. Commonly used for slow-moving organisms like mollusks, sponges, and benthic organisms in marine environments. Beat mark recapture method for mobile animals. Used for estimating populations of animals that move frequently, such as mammals, birds, and insects. Steps involve: capture a sample of individuals, mark them, and release them back into the population. After a period, recapture individuals from the population. Use the Lincoln-Peterson index to estimate population size, n equals m times c r n equals r m times c, where n equals estimated population size m equals number of marked individuals initially released c equals total number of individuals captured in second sampling r equals number of marked individuals recaptured assumes no births, deaths, immigration, or emigration between sampling events. Line transect method. Suitable for large mammals and birds in open landscapes. Involves walking along a straight line transect and recording animals observed within a fixed distance on either side. Distance sampling techniques can be applied to estimate density from these observations. Point count method. Commonly used for estimating bird and insect populations. Observers record individuals seen or heard from a fixed point within a set time period. 2. Indirect methods. These methods estimate population density using signs of animal presence rather than direct observation. Nest or burrow counts. Used for species that use nests or burrows, such as rodents, birds, and reptiles. The number of active nests or burrows within a given area is used to estimate population density. Beat, track, and sign surveys. Footprints, droppings, scratch marks, or other signs of activity provide estimates of population density, especially for elusive species. Camera trapping. Automated cameras placed in strategic locations capture images of animals, allowing density estimation through capture-recapture analysis. Frequently used for cryptic or nocturnal species like big cats and other mammals. Acoustic monitoring. Used for species that communicate via sound, such as birds, frogs, and bats. Population density is estimated based on the frequency of vocalizations recorded within a specified area. Methods for estimating population density of plants. 1. Quadrat sampling. The most widely used method for plants. A quadrat is placed randomly or systematically within the study area, and the number of individuals is counted. Density calculation. Density equals total number of individuals counted total area sampled density equals total area sampled total number of individuals counted. Quadrats can be square, rectangular, or circular, depending on the vegetation type. 2. Line and belt transects. Line transect. A measuring tape is laid across a habitat, and plants touching or intersecting the line are recorded. Belt transect. Similar to line transect but covers a wider strip of land, increasing accuracy for species occurring in clusters. 3. Point-centered quarter method. Used in forest ecology to estimate tree density. A series of random points are selected, and the nearest individual in each of 4 quadrants around the point is recorded. Density is estimated using D equals NAD equals AN, where N is the total count of plants in A is the area sampled. 4. Remote sensing and GIS techniques. Satellite imagery and aerial photography help estimate vegetation cover and density over large areas. Commonly used in forest and landscape ecology to assess biodiversity and habitat changes. Importance of estimating population density. Conservation biology helps assess the status of endangered species. Ecosystem management informs decisions on sustainable harvesting and habitat restoration. Ecological research provides insights into species interactions and community dynamics. Biodiversity monitoring tracks population trends over time, crucial for climate change impact assessment. Previous year solved question. Question. Which of the following is the most suitable method for estimating the population density of a highly mobile animal species? A. Quadrat sampling. B. Mark recapture method. C. Belt transect method. D. Point-centered quarter method. Answer. Correct option, B. Mark recapture method. Explanation. Highly mobile species cannot be effectively counted using quadrats or transects because they move in and out of the sampled area. The mark recapture method allows estimation of population size based on the proportion of marked individuals recaptured in subsequent samples. This concludes the comprehensive guide on methods of estimating population density. Let me know the next topic, and I will include MCQs as well. You said. Ranging patterns through direct. ChatGPT said. Ranging patterns through direct methods. Introduction. Ranging patterns refer to the movement and spatial distribution of animals within their habitat. These patterns are influenced by factors such as resource availability, predation risk, social structure, and reproductive behavior. Understanding ranging patterns is crucial for conservation biology, wildlife management, and behavioral ecology. Direct methods for studying ranging patterns involve direct observation and tracking of animals within their natural habitat. Direct methods for studying ranging patterns. 1. Direct observation. One of the simplest methods, where researchers visually follow and record an animal's movement. Best suited for large, diurnal, and easily observable species like primates, ungulates, and large carnivores. Observers use binoculars, spotting scopes, and field notebooks to document movement patterns, resting sites, feeding areas, and interactions. Limitations: observer presence may alter animal behavior, and it is challenging for nocturnal or cryptic species. 2. Radio telemetry (VHF) tracking. A widely used method where a small radio transmitter is attached to the animal via collar, harness, or implant. The researcher uses a directional antenna and receiver to detect the signal and determine the animal's location. This method provides precise movement data and helps study home range, habitat use, and migration. Limitations: requires frequent field tracking, and the signal range is limited by terrain and vegetation. 3. GPS tracking. Advanced technology using GPS collars or tags that provide real-time data on animal movement. Provides highly accurate location data, even for species that move over vast areas. Data can be transmitted via satellite or stored for later retrieval. Useful for studying long-distance migrations, habitat preferences, and responses to environmental changes. Limitations: expensive, requires battery maintenance, and may affect animal behavior if the device is too large. 4. Direct marking and resighting. Involves marking animals with paint, dyes, bands, or tags and tracking their movement based on sightings. Commonly used for birds banding, amphibians, toe clipping, and insects wing marking. Helps estimate dispersal, site fidelity, and territory shifts. Limitations: requires repeated resightings, and markings may fade or get lost over time. 5. Camera trapping. Motion-activated cameras placed in strategic locations to record images or videos of animals as they move through an area. Effective for monitoring elusive, nocturnal, or large-ranging species. Data can be used to map movement corridors and identify preferred routes. Limitations: limited to areas where cameras are placed, and detection rates depend on species behavior. 6. Drone surveillance. Use drones equipped with high-resolution cameras or thermal imaging to track animal movements over large landscapes. Useful for monitoring large herbivores, marine mammals, and migratory birds. Limitations: weather conditions can affect drone performance, and some animals may react to drone noise. Importance of studying ranging patterns. Conservation planning helps in designing protected areas and wildlife corridors. Human-wildlife conflict mitigation identifies conflict zones where animals interact with human settlements. Behavioral ecology provides insights into social organization, mating strategies, and foraging behavior. Epidemiology helps track disease spread among animal populations. Previous year solved question. Question: which of the following is the most suitable method for studying the ranging pattern of a migratory bird species? A. Direct observation. B. Mark resighting. C. Radio telemetry. D. GPS tracking. Answer: correct option, D. GPS tracking. Explanation: GPS tracking provides precise and continuous location data, making it ideal for studying long-distance migrations. Other methods like direct observation and mark resighting are limited in scope for highly mobile species. Multiple choice questions MCQs. 1. Which of the following direct methods provides the most precise data on animal movement patterns? A. Visual observation. B. Camera trapping. C. GPS tracking. D. Mark resighting. Answer: C. GPS tracking. 2. Which method is commonly used for tracking large mammals in dense forests? A. Drone surveillance. B. VHF radio telemetry. C. Direct observation. D. Paint marking. Answer: B. VHF radio telemetry. 3. Camera trapping is least effective for tracking at nocturnal species. B. Arboreal species. C. Large carnivores. D. Ground-dwelling mammals. Answer: B. Arboreal species. This concludes the detailed lecture on ranging patterns through direct methods. Let me know the next topic. You said, indirect, ChatGPT said. Ranging patterns through indirect methods. Introduction. Ranging patterns refer to how animals move within their habitat, including their home range, territoriality, and migration routes. When direct observation is difficult due to an animal's elusive nature, nocturnal behavior, or inaccessible habitats, indirect methods are used. These methods rely on physical evidence left by animals rather than direct tracking, making them essential for studying cryptic, rare, or wide-ranging species. Indirect methods for studying ranging patterns. 1. Track and sign surveys. The method involves identifying and analyzing footprints, feces, scratch marks, nests, burrows, feeding sites, and other signs of an animal's presence. Used extensively for mammals such as big cats, bears, and ungulates. Example: tiger pug marks paw prints are used in India to estimate population density and movement patterns. Limitations: requires expertise in track identification, and environmental conditions rain, wind may erase tracks. 2. Scat and pellet analysis. The location and frequency of fecal matter provide insights into an animal's movement, diet, and territorial behavior. Used for carnivores e.g., wolves, leopards, and herbivores e.g., deer, rabbits. DNA extracted from fecal samples helps identify individual animals and track migration. Limitations: decomposition and environmental factors may make scat identification difficult. 3. Hair and feather collection. Shed hair and feathers help determine the presence and movement of species, particularly in dense forests. Hair snares, sticky traps, or barbed wire are placed in animal pathways to collect samples. Example: used to study bears and large felids. Limitations: requires DNA analysis for species identification, and collection rates can be low. 4. Acoustic monitoring. Used for species that communicate through sound, such as birds, bats, whales, and amphibians. Recorders are placed in habitats to detect vocalizations and analyze movement patterns. Example: bat echolocation calls are used to determine flight paths and feeding ranges. Limitations: requires advanced software to analyze vocalizations and distinguish between species. 5. Camera trapping is an indirect method. Unlike direct tracking, camera traps passively record animal presence at specific locations. By analyzing capture locations and frequencies, researchers infer movement corridors. Used widely for species like tigers, leopards, and other nocturnal mammals. Limitations: detection depends on camera placement, and some animals may avoid cameras. 6. Stable isotope analysis. Analyzes chemical signatures in hair, bones, or feathers to infer an animal's geographic movements. Different geographic regions have unique isotopic compositions, allowing researchers to trace migration patterns. Example, used in bird migration studies. Limitations, requires specialized laboratory equipment and expertise. 7. Genetic and environmental DNA analysis. DNA from hair, scat, saliva, or environmental samples e.g., water, soil helps track an animal's presence and movement. Example, aquatic species like fish and amphibians are tracked using water samples. Limitations, requires molecular biology expertise and may not provide real-time movement data. Importance of indirect methods in studying ranging patterns non-intrusive, does not disturb or alter animal behavior. Effective for elusive species, useful for nocturnal, rare, or cryptic species that avoid human presence. Cost-effective, many methods require minimal equipment compared to GPS tracking. Long-term monitoring, provides data over extended periods without requiring continuous human presence. Previous year solved question. Question. Which of the following indirect methods is commonly used to study the ranging patterns of large carnivores in dense forests? A. GPS tracking. B. Scat analysis. C. Direct observation. D. Radio telemetry. Answer. Correct option, B. Scat analysis. Explanation. Large carnivores like tigers and leopards are difficult to observe directly in dense forests. Scat analysis helps infer movement patterns, diet, and territory range based on collected fecal samples. Multiple choice questions MCQs. 1. Which of the following methods is best suited for tracking bat movement patterns? A. Camera trapping. B. Acoustic monitoring. C. Direct observation. D. Mark recapture. Answer, B. Acoustic monitoring. 2. Environmental DNA is particularly useful for tracking the movement of arboreal mammals. B. Aquatic species. C. Large carnivores. D. Nocturnal birds. Answer, B. Aquatic species. 3. Which of the following is an indirect method that requires laboratory analysis for species identification? A. GPS tracking. B. Stable isotope analysis. C. Point transect method. D. Direct marking. Answer, B. Stable isotope analysis. This concludes the lecture on ranging patterns through indirect methods. Let me know the next topic. You said, remote observations. ChatGPT said. Remote observations in studying animal ranging patterns. Introduction. Remote observation methods allow researchers to study the movement and behavior of animals without direct interaction. These methods are crucial for monitoring elusive, migratory, or dangerous species across large landscapes. By using advanced technologies, researchers can collect accurate data on home range, habitat use, and migration patterns without disturbing the animals. Types of remote observation methods. 1. GPS and satellite tracking. Technology used: GPS collars, satellite tags, and geolocators. How it works: devices continuously record an animal's location and transmit data via satellites. Applications: tracking migratory birds, marine mammals, and large terrestrial species. Identifying movement corridors and conservation priorities. Example, satellite tracking of sea turtles to study migration routes. Limitations: high cost, battery constraints, and potential impact on animal behavior. 2. Camera traps. Remote sensing cameras. Technology used: motion-activated cameras with infrared night vision capabilities. How it works: cameras placed in strategic locations capture images, videos when triggered by movement. Applications: studying species distribution, habitat use, and behavior. Detecting cryptic or nocturnal animals like leopards and pangolins. Example, camera traps used in the Western Ghats to study tiger movement. Limitations: requires proper placement, and some species may avoid cameras. 3. Acoustic and bioacoustic monitoring. Technology used: autonomous recording units, ARIS, hydrophones for underwater species. How it works: devices record species-specific sounds like vocalizations, echolocation calls, and songs. Applications: studying bat flight paths, bird migration, and marine mammal communication. Identifying species presence in dense forests or deep ocean. Example, hydrophones track whale migrations by detecting their calls. Limitations: requires sophisticated software for sound analysis and species identification. 4. Drone UAV surveillance. Technology used: unmanned aerial vehicles, UAVs, with high-resolution cameras or thermal imaging. How it works: drones capture aerial footage of animal movements over large areas. Applications: monitoring large herbivores, elephants, zebras in savannas. Detecting poaching activities in protected areas. Example, drones used in Africa for anti-poaching efforts. Limitations: weather conditions affect performance, and some animals react to drone noise. 5. Remote sense GIS geographic information system mapping technology used: satellite imagery, lidar, GIS software. How it works: satellite data is analyzed to map habitat use and landscape changes. Applications: tracking habitat loss and fragmentation. Studying migratory patterns of birds using environmental variables. Example, GIS mapping used to analyze elephant corridors in India. Limitations: requires expertise in remote sensing analysis. 6. Environmental DNA analysis. Technology used: water, soil, or air samples analyzed for traces of genetic material. How it works: collected samples contain DNA shed by animals, which is then sequenced in a lab. Applications: detecting aquatic species like fish, amphibians, and otters. Identifying the presence of cryptic terrestrial animals. Example, EDNA used to track endangered amphibians in rainforest streams. Limitations: requires laboratory analysis and does not provide real-time tracking. Advantages of remote observations: non-intrusive, no direct human-animal interaction, reducing behavioral disturbances. Long-term monitoring, allows continuous data collection over months or years. Global coverage, enables the study of migratory and deep-sea species. Climate resilience, useful for monitoring species in inaccessible or extreme environments. Previous year solved question. Question: which of the following remote observation methods is most effective for tracking migratory patterns of large marine animals? A. Camera trapping. B. GPS and satellite tracking. C. Acoustic monitoring. D. Mark recapture. Answer: correct option, B. GPS and satellite tracking. Explanation. GPS and satellite tags provide real-time, long-distance movement data, making them ideal for tracking marine animal migrations. Other methods like camera trapping and acoustic monitoring are location-dependent. Multiple choice questions MCQs. 1. Which method is most suitable for studying the movement of nocturnal species in dense forests? A. GPS tracking. B. Camera trapping. C. Drone surveillance. D. EDNA analysis. Answer. B. Camera trapping. 2. Which of the following is a major limitation of using drone surveillance for tracking animal movement? A. High cost. B. Poor accuracy. C. Short battery life and weather dependence. D. Inability to capture nocturnal activity. Answer. C. Short battery life and weather dependence. 3. Which remote sensing technology is widely used to map animal habitats and migration routes? A. LiDAR and GIS. B. Acoustic monitoring. C. Radio telemetry. D. Track surveys. Answer. A. LiDAR and GIS. This concludes the lecture on remote observations and studying ranging patterns. Let me know the next topic. You said. Sampling methods in the study of behavior. ChatGPT said. Sampling methods in the study of behavior. Introduction. Behavioral research involves observing and recording the actions of animals in different contexts, such as foraging, mating, aggression, and communication. Since it is impractical to observe all behaviors at all times, researchers use specific sampling methods to systematically collect data. The choice of method depends on the research question, the species being studied, and the environment. Types of sampling methods. 1. Ad libitum sampling, unstructured observation. Description. The observer records any and all behaviors of interest without any systematic constraints. Application. Useful for preliminary studies to identify key behaviors. Often used in ethological research on social animals like primates. Limitations. Observations may be biased toward conspicuous behaviors. Not suitable for quantitative comparisons. 2. Focal animal sampling. Description. Observing and recording the behavior of a single individual for a specified period. Application. Studying detailed behavior of an individual, e.g., dominance interactions, grooming frequency in primates. Used in long-term behavioral studies. Example. Observing a single chimpanzee for an hour to record grooming, eating, and aggression. Limitations. Requires clear individual identification. May miss social interactions occurring outside the focal animal's range. 3. Scan sampling, instantaneous sampling. Description. Observing and recording the behavior of an entire group at regular time intervals. Application. Efficient for studying social structures and group activity budgets. Common in bird flock and primate troop studies. Example. Every 10 minutes, recording how many individuals are feeding, resting, or interacting in a monkey troop. Limitations. Less detailed information on individual behavior. Short duration behaviors, e.g., quick fights, may be missed. 4. Behavior sampling, event sampling. Description. Recording specific behaviors whenever they occur, regardless of which individual performs them. Application. Used for rare but significant behaviors, e.g., mating, aggression, alarm calls. Suitable for studying infrequent events like predation attempts. Example. Recording each time a lion roars or a male peacock displays his feathers. Limitations. Observer fatigue can lead to missed events. Does not provide continuous behavioral data. 5. Continuous sampling, all occurrences sampling. Description. Every instance of a particular behavior is recorded for a set time. Application. Ideal for studying high-frequency behaviors, e.g., grooming, play behavior. Provides detailed quantitative data. Example. Recording every grooming event in a monkey group for 1 hour. Limitations. Requires intense concentration and effort. Not feasible for long observation periods. 6. 1-0 sampling, time sampling method. Description. Behavior is recorded as either present 1 or absent 0 within a set time interval, rather than counting exact occurrences. Application. Simplifies data collection in fast-moving or large groups. Used in studies of group behavior in birds, fish, and mammals. Example. Every 5 minutes, recording whether or not an animal is engaged in play behavior. Limitations. Does not account for duration or frequency of behavior. Can underestimate actual behavior occurrence. Choosing the right sampling method. For social behavior, focal animal sampling or scan sampling is effective. For rare behaviors, behavior sampling is best. For general observations, ad libitum sampling is useful. For quantitative behavioral analysis, continuous sampling provides detailed data. Previous year solved question. Question. Which sampling method is best suited for studying infrequent but significant behaviors, such as mating displays or predator attacks? A. Scan sampling. B. Focal animal sampling. C. Behavior sampling. D. Continuous sampling. Answer. Correct option, C. Behavior sampling. Explanation. Behavior sampling is ideal for recording rare behaviors, as it focuses on specific events whenever they occur, rather than on individuals or time intervals. Multiple choice questions MCQs. 1. Which sampling method is best for studying the overall activity budget of a primate group? A. Behavior sampling. B. Scan sampling. C. Continuous sampling. D. 1-0 sampling. Answer. B. Scan sampling. 2. In which sampling method does an observer record everything a single individual does for a set period? Sampling methods in the study of behavior. ChatGPT said. Sampling methods in the study of behavior. Introduction. Behavioral research involves observing and recording the actions of animals in different contexts, such as foraging, mating, aggression, and communication. Since impractical to observe all behaviors at all times, researchers use specific sampling methods to systematically collect data. The choice of method depends on the research question, the species being studied, and the environment. Types of sampling methods. 1. Ad libitum sampling, unstructured observation. Description. The observer records any and all behaviors of interest without any systematic constraints. Application. Useful for preliminary studies to identify key behaviors. Often used in ethological research on social animals like primates. Limitations. Observations may be biased toward conspicuous behaves. Not suitable for quantitative comparisons. 2. Focal animal sampling. Types of sampling methods. 1. Ad libitum sampling, unstructured observation. Description. The observer records any and all behaviors of interest without any systematic constraints. Application. Useful for preliminary studies to identify key behaviors. Often used in ethological research on social animals like primates. Limitations. Observations may be biased toward conspicuous behaviors. Not suitable for quantitative comparisons. 2. Focal animal sampling. Description. Observing and recording the behavior of a single individual for a specified period. Application. Studying detailed behavior of an individual, e.g., dominance interactions, grooming frequency in primates. Used in long-term behavioral studies. Example. Observing a single chimpanzee for an hour to record grooming, eating, and aggression. Limitations. Requires clear individual identification. Name is social interactions occurring outside the focal animal's range. 3. Scan sampling, instantaneous sampling. Description. Observing and recording the behavior of an entire group at regular time intervals. Application. Efficient for studying social structures and group activity butts. Common in bird flock and primate troop studies. Example. Every 10 minutes, recording how many individuals are feeding, resting, or interacting in a monkey troop. Limitations. Less detailed information on individual behavior. Short duration behaviors, e.g., quick fights may be missed. 4. Behavior sampling, event sampling. Description. Recording specific behaviors whenever they occur, regardless of which individual performs them. Application. Used for rare but significant behaviors, e.g., mating, aggression, alarm calls. Suitable for studying infrequent events like predation attempts. Example. Recording each time a lion roars or a male peacock displays his feathers. Limitations. Observer fatigue can lead to missed events. Does not provide continuous behavioral data. 5. Continuous sampling, all occurrences sampling. Description. Every instance of a particular behavior is recorded for a set time. Application. Ideal for studying high-frequency behaviors, e.g., grooming, play behavior. Provides detailed quantitative data. Example. Recording every grooming event in a monkey group for 1 hour. Limitations. Requires intense concentration and effort. Not feasible for long observation periods. 6. 1-0 sampling, time sampling method. Description. Behavior is recorded as either present 1 or absent 0 within a set time interval, rather than counting exact occurrences. Application. Simplifies data collection in fast-moving or large groups. Used in studies of group behavior in birds, fish, and mammals. Example. Every 5 minutes, recording whether or not an animal is engaged in play behavior. Limitations. Does not account for duration or frequency of behavior. Can underestimate actual behavior occurrence. Choosing the right sampling method. For social behavior, focal animal sampling or scan sampling is effective. For rare behaviors, behavior sampling is best. For general observations, ad libitum sampling is useful. For quantitative behavioral analysis, continuous sampling provides detailed data. Previous year solved question. Question. Which sampling method is best suited for studying infrequent but significant behaviors, such as mating displays or predator attacks? A. Scan sampling. B. Focal animal sampling. C. Behavior sampling. D. Continuous sampling. Answer. Correct option, C. Behavior sampling. Explanation. Behavior sampling is ideal for recording rare behaviors, as it focuses on specific events whenever they occur, rather than on individuals or time intervals. Multiple choice questions, MCQs. 1. Which sampling method is best for studying the overall activity budget of a primate group? A. Behavior sampling. B. Scan sampling. C. Continuous sampling. D. 1-0 sampling. Answer. B. Scan sampling. 2. In which sampling method does an observer record everything a single individual does for a set period? A. Focal animal sampling. B. Ad libitum sampling. C. Behavior sampling. D. 1-0 sampling. Answer. A. Focal animal sampling. 3. 1-0 sampling is most useful when A researcher needs to quantify precise behavior durations. B. Observing behaviors that occur frequently in a group. C. Rare behaviors need to be recorded systematically. D. Studying territorial disputes in solitary carnivores. Answer. B. Observing behaviors that occur frequently in a group. This concludes the lecture on sampling methods in the study of behavior. Let me know the next topic. You said. Habitat characterization. ChatGPT said. Habitat characterization introduction. A habitat is a natural environment where an organism or a population lives, providing the necessary resources for survival, growth, and reproduction. Habitat characterization is the process of describing and analyzing the physical, chemical, and biological attributes of a habitat to understand its suitability for different species. This is a fundamental aspect of ecological studies, conservation biology, and wildlife management. Components of habitat characterization. 1. Physical characteristics. These include abiotic factors that define the structure and conditions of a habitat. A. Climate and microclimate. Temperature influences metabolism, breeding cycles, and species distribution. Humidity affects water balance and thermoregulation in animals. Precipitation determines water availability, which is crucial for plants and dependent fauna. Wind patterns impact seed dispersal, evaporation rates, and soil erosion. B. Topography and geomorphology. Altitude and elevation. Higher altitudes often have lower temperatures and oxygen levels. Slope and aspect affect sunlight exposure, water drainage, and soil stability. Soil type and composition determines nutrient availability, water retention, and vegetation growth. C. Water availability. Presence of rivers, lakes, and wetlands, essential for aquatic and semi-aquatic organisms. Groundwater levels influence plant root systems and soil moisture. pH and salinity affect species that rely on freshwater or marine habitats. 2. Chemical characteristics. These determine the quality of the habitat in terms of nutrient cycles and pollution levels. A. Soil chemistry. pH levels, influence plant growth and microbial activity. Nutrient content N, P, K essential for plant productivity and food chain stability. Organic matter, supports soil fertility and microbial life. B. Water chemistry. Dissolved oxygen DO, critical for aquatic organisms respiration. Nutrient load, eutrophication, excess nitrogen and phosphorus can lead to algal blooms. Toxic contaminants, heavy metals and pesticides impact biodiversity. 3. Biological characteristics. These include the living components of a habitat that interact with the abiotic environment. A. Vegetation structure. Floral diversity, determines the primary productivity of an ecosystem. Canopy cover, influences light penetration and understory plant growth. Litter and deadwood, provide microhabitats for decomposers and invertebrates. B. Faunal composition. Keystone species, play a critical role in maintaining ecosystem balance, e.g., predators, pollinators. Indicator species, signal environmental health, e.g., amphibians indicate water quality. Trophic interactions, predator-prey and herbivore-plant relationships structure communities. C. Microbial communities. Decomposers, fungi and bacteria that recycle nutrients. Symbiotic associations, mycorrhizae and plant roots enhance nutrient uptake. Methods of habitat characterization. 1. Vegetation sampling. Quadrat method, used for estimating plant species richness and abundance. Transect method, a line is drawn across a habitat, and vegetation along it is recorded. Canopy cover estimation, determines the amount of sunlight reaching the ground. 2. Soil and water analysis. Soil sampling, pH, texture, and nutrient analysis. Water sampling, dew, salinity, and pollutant measurement. 3. Faunal surveys. Camera trapping, records animal presence and movement. Point counts and line transects, used for bird and mammal surveys. Pitfall traps and sweep nets, capture insects and small ground-dwelling species. 4. Remote sensing and GIS. Satellite imagery, monitors habitat changes over time. GIS mapping, creates spatial distribution maps of species and environmental variables. Importance of habitat characterization. Biodiversity conservation, identifies critical habitats for protection and restoration. Climate change impact assessment, helps track shifts in habitat conditions. Wildlife management, guides habitat improvement projects for endangered species. Ecosystem monitoring, detects degradation and guides mitigation strategies. Previous year solved question. Question. Which of the following methods is best suited for assessing plant species diversity in a forest habitat? A. Camera trapping. B. Quadrat sampling. C. Pitfall traps. D. Radio telemetry. Answer. Correct option, B. Quadrat sampling. Explanation. Quadrat sampling is a widely used method in vegetation studies for estimating species richness, density, and cover. Other methods listed are used for faunal studies. Multiple choice questions MCQs. 1. Which of the following factors is not an abiotic characteristic of a habitat? A. Soil pH. Canopy cover. C. Precipitation. D. Wind speed. Answer. B. Canopy cover. It is a biotic characteristic. 2. Which method is most effective for assessing large-scale habitat changes over time? A. GIS and remote sensing. B. Pitfall traps. C. Line transects. D. Quadrat sampling. Answer. A. GIS and remote sensing. 3. Why are indicator species important in habitat characterization? A. They are the most abundant species in an area. B. They indicate the presence of predators. C. They reflect environmental health and habitat quality. D. They are always at the top of the food chain. Answer. C. They reflect environmental health and habitat quality. This concludes the lecture on habitat characterization. Let me know the next topic. He said. Ground and remote sensing methods. ChatGPT said. Ground and remote sensing methods in habitat characterization introduction. Habitat characterization requires accurate and reliable methods to assess various environmental parameters. Researchers use ground-based methods for detailed, localized studies and remote sensing techniques for broad-scale habitat analysis. The combination of these approaches helps in understanding habitat quality, biodiversity patterns, and environmental changes. Ground-based methods. Ground-based methods involve direct field observations and measurements. These techniques provide high-resolution, accurate data but are often time-consuming and limited in spatial coverage. 1. Vegetation surveys. Quadrat sampling, a fixed area frame e.g., 1 square meter or 10 square meters, is used to estimate plant diversity, density, and abundance. Line transects, a straight line is laid out, and vegetation data is recorded at intervals along it. Point-centered quarter method, used to estimate tree density by measuring distances between trees at random points. 2. Soil and water analysis. Soil sampling, collection of soil samples at various depths for analysis of pH, nutrient content, and moisture. Water quality testing, measurement of dissolved oxygen DO, pH, salinity, turbidity, and heavy metals to assess aquatic habitats. 3. Faunal monitoring techniques. Camera traps, motion sensor cameras record wildlife presence and behavior. Pitfall traps, small containers placed in the ground capture insects, amphibians, and small mammals. Mist nets, used to capture and study birds and bats. Acoustic monitoring, records vocalizations of birds, bats, and amphibians to study species diversity. 4. Microclimate measurements. Temperature and humidity sensors, used to monitor variations in local climate conditions. Light intensity meters, measure solar radiation penetration in forest ecosystems. Anemometers, record wind speed and direction. Remote sensing methods. Remote sensing involves collecting data from a distance, typically using satellites, drones, and aerial surveys. These methods allow for large-scale, repeated measurements of habitat characteristics. 1. Satellite remote sensing. Optical remote sensing, uses visible and infrared sensors to assess vegetation cover, land use changes, and habitat fragmentation. Radar remote sensing, uses microwave signals to penetrate clouds and map forest structure, water bodies, and landforms. Litter light detection and ranging, uses laser pulses to create 3D models of forests, canopy heights, and terrain elevations. 2. UAV drone surveys. High-resolution imaging, drones equipped with cameras capture detailed habitat maps. Multispectral and thermal sensors, detect plant health, animal movement, and water stress. 3D habitat modeling, generates topographic models to study species distribution. 3. Geographic information systems, GIS. Habitat mapping, integrates satellite and ground data to create spatial models. Change detection, monitors deforestation, wetland degradation, and urban expansion over time. Species distribution models, predicts potential habitats based on environmental variables. 4. Aerial photography. Fixed-wing aircraft and helicopters, used for high-resolution imaging of inaccessible regions. Thermal imaging detects heat signatures of animals, useful for nocturnal species studies. Comparison of ground and remote sensing methods. Aspect ground-based methods. Remote sensing methods. Scale of study. Local detailed analysis. Large-scale habitat mapping. Accuracy. Height. Site-specific. Moderate. Dependent on resolution. Cost and time. Labor-intensive. Costly for large areas. Cost-effective for large-scale studies. Environmental impact can be invasive. Trapping. Sampling. Non-invasive. Satellite. Drone surveys. Best used for soil, microclimate, species interactions. Land cover. Habitat change. Large-scale mapping. Applications of ground and remote sensing methods. Biodiversity conservation. Identifying critical habitats for endangered species. Climate change studies. Monitoring temperature, vegetation shifts, and carbon sequestration. Ecosystem management. Tracking deforestation, wetland loss, and desertification. Disaster monitoring, assessing habitat destruction due to floods, fires, and landslides. Previous year solved question. Question. Which remote sensing technique is best suited for creating a 3D model of forest canopy height? A. Optical imaging. B. Lidar. C. Thermal sensing. D. Radar imaging. Answer. Correct option, B. Lidar. Explanation. Lidar uses laser pulses to generate high-resolution 3D models, making it the best technique for measuring canopy height and terrain elevation. Multiple choice questions. MCQs. 1. Which of the following is a ground-based method for habitat characterization? A. Lidar. B. Satellite imaging. C. Quadrat sampling. D. GIS mapping. Answer. C. Quadrat sampling. 2. What is the advantage of using UAVs, drones, in habitat studies? A. They can cover vast areas quickly with high-resolution data. B. They are limited to soil sampling only. C. They do not require any calibration. D. They replace all ground-based methods. Answer. A. They can cover vast areas quickly with high-resolution data. 3. Remote sensing methods are particularly useful for A. Measuring soil pH at different depths. B. Observing animal interactions in the field. C. Detecting land use changes over time. D. Capturing individual plant root growth. Answer. C. Detecting land use changes over time. This concludes the lecture on ground and remote sensing methods. Let me know the next topic. You said, biomolecules. 1. Structural composition. ChatGPT said. Biomolecules, structural composition introduction. Biomolecules are the fundamental chemical compounds that make up living organisms, playing crucial roles in cellular structure, metabolism, and function. They can be broadly classified into carbohydrates, proteins, lipids, and nucleic acids, each with distinct structural compositions and biological functions. The arrangement of atoms in these molecules determines their physical and chemical properties, influencing life at the molecular level. 1. Carbohydrates, structure and composition. Carbohydrates are organic molecules composed of carbon C, hydrogen H, and oxygen O in the general formula CHO. They serve as energy sources, structural components, and signaling molecules in cells. A. Monosaccharides, simple sugars, structure, single sugar units with a backbone of 3 to 7 carbon atoms. Examples. Glucose CHO, primary energy source. Fructose CHO, found in fruits. Ribose CHO, a component of RNA. B. Disaccharides. Structure, formed by the condensation of 2 monosaccharides via a glycosidic bond. Examples. Sucrose, glucose plus fructose, table sugar. Lactose, glucose plus galactose, milk sugar. C. Polysaccharides. Structure, long chains of monosaccharides joined by glycosidic bonds. Examples. Starch, storage polysaccharide in plants. Glycogen, storage form of glucose in animals. Cellulose, structural component in plant cell walls. 2. Proteins, structure and composition. Proteins are polymers of amino acids, containing carbon C, hydrogen H, oxygen O, nitrogen N, and sometimes sulfur S. They play structural, enzymatic, transport, and regulatory roles in cells. A. Amino acid structure. Each amino acid has a common structure. A central carbon, alpha carbon, an amino group, NH, a carboxyl group, COOH. A variable R group, side chain, that determines chemical properties. B. Peptide bond formation. Amino acids are linked by peptide bonds, forming polypeptides. A dehydration reaction between the amino and carboxyl groups of adjacent amino acids results in a covalent bond. C. Protein structural organization. Primary structure, linear sequence of amino acids. Secondary structure, alpha helices and beta sheets formed by hydrogen bonding. Tertiary structure, 3-dimensional folding due to interactions among R groups. Quaternary structure, complex of multiple polypeptide chains, e.g., hemoglobin. 3. Lipids, structure and composition. Lipids are hydrophobic molecules made of carbon C, hydrogen H, and oxygen O, with some containing phosphorus P and nitrogen N. They function as energy reserves, membrane components, and signaling molecules. A. Fatty acids. Structure, hydrocarbon chains with a terminal carboxyl COO group. Types. Saturated fatty acids, no double bonds, e.g., palmitic acid. Unsaturated fatty acids, 1 or more double bonds, e.g., oleic acid. B. Triglycerides. Structure, composed of 1 glycerol molecule linked to 3 fatty acids via ester bonds. Function, major energy storage molecules in animals. C. Phospholipids. Structure, composed of a glycerol backbone, 2 fatty acids, and a phosphate group. Function, major components of biological membranes. D. Steroids. Structure, 4 interconnected carbon rings. Examples. Cholesterol, precursor for steroid hormones. Testosterone and estrogen, regulate reproductive functions. 4. Nucleic acids, structure and composition. Nucleic acids are polymers of nucleotides, composed of carbon C, hydrogen H, oxygen O, nitrogen N, and phosphorus B. They store and transmit genetic information. A. Nucleotide structure. Each nucleotide consists of a nitrogenous base, purines, adenine, guanine, pyrimidines, cytosine, thymine, uracil, a pentose sugar, deoxyribose DNA, ribose RNA, a phosphate group, B, DNA deoxyribonucleic acid. Structure, double-stranded helix with complementary base pairing, AT, GC. Function, stores genetic instructions for protein synthesis. C, RNA ribonucleic acid. Structure, single-stranded molecule. Types. mRNA, messenger RNA, carries genetic code from DNA to ribosomes. tRNA, transfer RNA, brings amino acids during protein synthesis. rRNA, ribosomal RNA, forms ribosomes for translation. Previous year solved question. Which biomolecule is the primary structural component of cell membranes? A. Proteins. B. Carbohydrates. C. Phospholipids. D. Nucleic acids. Answer. Correct option, C. Phospholipids. Explanation. Phospholipids form the bilayer of cell membranes, providing structural integrity and selective permeability. Multiple choice questions MCQs 1. Which of the following is a polysaccharide? A. Glucose. B. Sucrose. C. Glycogen. D. Ribose. Answer. C. Glycogen. 2. Peptide bonds are found in which biomolecule? A. Proteins. B. Lipids. C. Carbohydrates. D. Nucleic acids. Answer. A. Proteins. 3. Which nitrogenous base is found in RNA but not in DNA? A. Thymine. B. Adenine. C. Cytosine. D. Uracil. Answer. D. Uracil. This completes the lecture on structural composition of biomolecules. Let me know the next topic. You said. Atoms, molecules and chemical bonds. ChatGPT said. Atoms, molecules, and chemical bonds. Introduction. Atoms are the fundamental building blocks of matter, combining to form molecules through chemical bonds. These interactions determine the structure, stability, and function of all biological molecules. Understanding atomic structure, molecular formation, and bonding mechanisms is crucial for comprehending biochemical processes at the cellular level. 1. Atomic structure. Atoms consist of 3 fundamental subatomic particles. Protons plus charge found in the nucleus determine atomic number Z. Neutrons neutral also in the nucleus contribute to atomic mass. Electrons charge orbit the nucleus in energy levels shells. A. Atomic number and mass number. Atomic number Z. Number of protons in an atom, e.g., carbon C has Z equals 6. Mass number 8. Sum of protons and neutrons, e.g., oxygen 16 has A equals 16. B. Electron configuration and orbitals. Electrons occupy energy levels K, L, M, N. Following the 2n squared rule, valence electrons, outermost shell electrons determine an atom's reactivity. Elements achieve stability by completing their valence shell octet rule. 2. Molecules and chemical bonds. Atoms form molecules by chemical bonding to achieve stable configurations. The type of bond formed depends on electronegativity differences in valence electrons. A. Covalent bonds. Definition. Sharing of electrons between atoms. Types. Single bond, 1 pair of shared electrons H, CH. Double bond, 2 pairs of shared electrons O, CO. Triple bond, 3 pairs of shared electrons N. Example, water HO forms polar covalent bonds, where oxygen pulls electrons more strongly than hydrogen. B. Ionic bonds. Definition. Transfer of electrons from one atom to another, forming charged ions. Example, sodium Na donates an electron to chlorine Cl, forming NaCl and NaCl, table salt. Properties, strong in solid state but dissociate in water, electrolytes. C. Hydrogen bonds. Definition, weak attraction between a hydrogen atom and an electronegative atom O, N, F. Example, DNA base pairing AT, GC and water cohesion HO molecules sticking together. D. Van der Waals forces. Definition, weak, temporary interactions due to electron movement. Example, gecko feet sticking to walls. E. Hydrophobic interactions. Definition, nonpolar molecules, lipids clustered together in aqueous environments. Example, formation of biological membranes, phospholipid bilayer. 3. Biological significance of chemical bonds. Protein structure, peptide bonds link amino acids, while hydrogen bonds stabilize secondary structures. DNA stability, hydrogen bonding holds double-stranded DNA together. Membrane formation, hydrophobic interactions create lipid bilayers. Enzyme function, covalent and non-covalent interactions help in substrate binding. Previous year solved question. Question. Which type of bond is responsible for holding the 2 strands of DNA together? A. Covalent bond. B. Hydrogen bond. C. Ionic bond. D. Van der Waals forces. Answer. Correct option, B. Hydrogen bond. Explanation. Hydrogen bonds between nitrogenous bases AT, GC provide structural stability to DNA. Multiple choice questions MCQs. 1. Which type of bond is the strongest under physiological conditions? A. Ionic bond. B. Covalent bond. C. Hydrogen bond. D. Van der Waals forces. Answer. B. Covalent bond. 2. The attraction between a slightly positive hydrogen atom and a highly electronegative atom is called a covalent bond. B. Ionic bond. C. Hydrogen bond. D. Hydrophobic interaction. Answer. C. Hydrogen bond. 3. What type of bond holds non-Cl together in NaCl? A covalent bond. B. Ionic bond. C. Hydrogen bond. D. Van der Waals forces. Answer. B. Ionic bond. This completes the lecture on atoms, molecules, and chemical bonds. Let me know the next topic. You said. Composition, structure, and function of different biomolecules, carbohydrates, ChatGPT said. Composition, structure, and function of carbohydrates. Introduction. Carbohydrates are a class of organic compounds composed primarily of carbon C, hydrogen H, and oxygen O. They are one of the 4 major biomolecules, alongside proteins, lipids, and nucleic acids. Carbohydrates serve as essential sources of energy, structural components, and signaling molecules in living organisms. They are classified based on their structural complexity, ranging from simple sugars to complex polysaccharides. 1. Composition and structure of carbohydrates. Carbohydrates follow the general formula CHO, where N is the number of carbon atoms in the molecule. The simplest form of carbohydrates is monosaccharides, which can be combined to form more complex molecules like disaccharides and polysaccharides. A. Monosaccharides, simple sugars. Monosaccharides are the most basic units of carbohydrates, consisting of a single sugar molecule. They are classified based on the number of carbon atoms they contain. Triose, 3 carbon atoms, e.g., glyceraldehyde. Pentose, 5 carbon atoms, e.g., ribose, deoxyribose. Hexose, 6 carbon atoms, e.g., glucose, fructose, galactose. Monosaccharides are further categorized as aldoses with an aldehyde group or ketoses with a ketone group. For example, glucose is an aldose, and fructose is a ketose. Structure. Monosaccharides can exist in 2 structural forms, open chain and cyclic forms. The cyclic form is more stable, with a hemiacetal or hemiketal bond forming between the hydroxyl group and the carbonyl group. B. Disaccharides. Disaccharides are formed by the condensation of 2 monosaccharides through a glycosidic bond, which involves the elimination of a water molecule. The 2 monosaccharides can be either the same or different. Examples of disaccharides: sucrose, glucose plus fructose, table sugar. Lactose, glucose plus galactose, milk sugar. Maltose, glucose plus glucose found in malt. Structure. Disaccharides are held together by alpha or beta glycosidic linkages, which differ in the orientation of the bond between the two monosaccharides. The bond type influences the digestion and breakdown of the disaccharide. C. Polysaccharides. Polysaccharides are large, complex carbohydrates composed of many monosaccharide units linked together. They can be linear or branched in structure. Examples of polysaccharides. Starch, a storage polysaccharide in plants, made up of glucose units. It consists of two components, amylose unbranched and amylopectin branched. Glycogen, a storage polysaccharide in animals, primarily found in the liver and muscles. It is similar to amylopectin but more highly branched. Cellulose, a structural polysaccharide in plants, made of glucose units linked by beta-1, 4 glycosidic bonds. Humans cannot digest cellulose due to the beta linkage. Chitin, found in the exoskeleton of arthropods, made of N-acetylglucosamine units. Structure. Polysaccharides are formed through the repeated formation of glycosidic bonds between monosaccharides. Their structure is either branched, e.g., glycogen, or linear, e.g., cellulose. The type of glycosidic bond and branching affects the stability and function of the polysaccharide. 2. Functions of carbohydrates. Carbohydrates perform a wide range of biological functions, which can be broadly categorized into energy storage, structural support, cell recognition, and biological roles in metabolism. A. Energy storage and supply. Carbohydrates are the primary energy source for living organisms. Glucose, a simple sugar, is the main energy source for cellular processes. When glucose is in excess, it is stored as glycogen in animals or starch in plants. Glycogen is rapidly mobilized to release glucose when the body requires energy, such as during exercise or between meals. Starch serves as an energy reserve in plants and is broken down into glucose during periods of metabolic demand. B. Structural function. Carbohydrates play crucial roles in providing structural support to cells and organisms. Cellulose provides rigidity to plant cell walls and is essential for maintaining plant structure. Chitin forms the exoskeleton of arthropods and provides protection and structural integrity. C. Cell signaling and recognition. Carbohydrates are involved in cell recognition and signaling, especially on the cell surface. They are attached to proteins forming glycoproteins or lipids forming glycolipids on the plasma membrane. These structures play a key role in immune recognition, tissue formation, and signaling pathways. Blood type. The different blood types A, B, O are determined by the specific carbohydrate structures attached to proteins on red blood cells. D. Biological roles in metabolism. Carbohydrates are essential for metabolic pathways like glycolysis, gluconeogenesis, and the pentose phosphate pathway, all of which involve the breakdown or synthesis of sugars for energy production and biosynthesis. Previous year solved question. Which of the following carbohydrates is a storage form of glucose in animals? A. Starch. B. Cellulose. C. Glycogen. D. Chitin. Answer. Correct option, C. Glycogen. Explanation. Glycogen is the storage form of glucose in animals, primarily stored in the liver and muscles. It can be rapidly broken down to release glucose when needed for energy. Multiple choice questions. MCQs 1. Which of the following is a disaccharide? A. Fructose. B. Sucrose. C. Glucose. D. Starch. Answer. B. Sucrose. 2. The structure of cellulose consists of alpha-1, 4 glycosidic bonds. B. Beta-1, 4 glycosidic bonds. C. Alpha-1, 6 glycosidic bonds. D. Beta-1, 6 glycosidic bonds. Answer. B. Beta-1, 4 glycosidic bonds. 3. Which carbohydrate is used by plants for energy storage? A. Starch. B. Glycogen. C. Cellulose. D. Chitin. Answer. A. Starch. This completes the lecture on carbohydrates, composition, structure, and function. Let me know the next topic. You said, lipids, ChatGPT said. Lipids, composition, structure, and function. Introduction. Lipids are a diverse group of hydrophobic or amphipathic biomolecules that play critical roles in energy storage, structural components of cell membranes, and signaling. They are primarily composed of carbon C, hydrogen H, and oxygen O, and in some cases, nitrogen N, and phosphorus B. Unlike carbohydrates and proteins, lipids are not soluble in water but are soluble in nonpolar solvents such as chloroform, ether, or acetone. Lipids are classified based on their structure and function, and they serve essential biological purposes, particularly in the organization and maintenance of cellular integrity. 1. Composition and structure of lipids. Lipids can be classified into simple lipids, compound lipids, and derived lipids based on their structure. The basic components of lipids are fatty acids and glycerol, though variations arise depending on the presence of other functional groups. A. Fatty acids. Fatty acids are long chains of hydrocarbons with a carboxyl group, Cu, at one end. They can be saturated, no double bonds between carbon atoms, or unsaturated, one or more double bonds between carbon atoms. The presence of double bonds in unsaturated fatty acids introduces kinks in the chain, affecting the fluidity of lipid structures. Saturated fatty acids. These have no double bonds and are typically solid at room temperature, e.g., stearic acid. Unsaturated fatty acids. These have one or more double bonds and are usually liquid at room temperature, e.g., oleic acid, linoleic acid. Monounsaturated. One double bond, e.g., oleic acid. Polyunsaturated. Multiple double bonds, e.g., linoleic acid, arachidonic acid. B. Glycerol and triglycerides. Glycerol is a 3-carbon molecule with hydroxyl O groups attached to each carbon. Triglycerides are the most common form of lipids used for energy storage. They are formed by esterification, where 3 fatty acids bond with one glycerol molecule, producing a triglyceride and releasing 3 molecules of water. Structure of triglycerides. They consist of a glycerol backbone with 3 fatty acid chains. Triglycerides are efficient energy storage molecules because they contain a high amount of chemical energy stored in the bonds of the fatty acids. C. Phospholipids. Phospholipids are a class of compound lipids that are a major component of cell membranes. They consist of a glycerol backbone, 2 fatty acid chains, hydrophobic tails, and a phosphate group, hydrophilic head. The amphipathic nature of phospholipids, having both hydrophilic and hydrophobic regions, allows them to form bilayers, which serve as the structural foundation of biological membranes. Example, phosphatidylcholine, a major component of cell membranes, has a choline group attached to the phosphate. D. Steroids. Steroids are a class of derived lipids that have a structure composed of 4 fused carbon rings. The most well-known steroid is cholesterol, a crucial component of cell membranes and a precursor to steroid hormones, e.g., estrogen, testosterone, cortisol, and bile acids. Structure of cholesterol. It consists of 4 fused carbon rings, a hydroxyl group at one end, and a hydrocarbon tail at the other. Cholesterol maintains membrane fluidity by preventing the fatty acid chains from packing too tightly in the membrane. E. Waxes. Waxes are esters of long-chain fatty acids and long-chain alcohols. They are hydrophobic and serve protective roles in organisms, such as waterproofing in plants e.g., cuticle and animal fur e.g., earwax. 2. Functions of lipids. Lipids play several vital roles in biological systems, ranging from energy storage to structural support to signaling. A. Energy storage. Lipids, particularly triglycerides, are the most energy-dense biomolecules, providing more than twice the energy per gram than carbohydrates or proteins. Triglycerides are stored in adipose tissue in animals, providing an efficient and long-term energy reserve. When energy is needed, triglycerides are broken down into fatty acids and glycerol and metabolized in the mitochondria to produce ATP. B. Structural role. Phospholipids form the core structure of biological membranes. The phospholipid bilayer is essential for maintaining the integrity of cells and organelles by providing a selective barrier that controls the passage of ions, molecules, and water. Cholesterol, embedded within the lipid bilayer, modulates membrane fluidity, ensuring stability under varying temperature conditions. C. Signaling molecules. Many lipids function as signaling molecules. Steroid hormones e.g., cortisol, estrogen, testosterone regulate various physiological processes such as metabolism, immune function, and reproduction. Eicosanoids, derived from polyunsaturated fatty acids like arachidonic acid, play roles in inflammation, immune responses, and blood clotting. D. Insulation and protection. Lipids, such as adipose tissue, serve as thermal insulators, helping organisms maintain body temperature. Additionally, lipids provide mechanical protection to vital organs e.g., kidney, heart by acting as shock absorbers. E. Waterproofing. Waxes are important in preventing water loss in organisms. In plants, the cuticle, a waxy coating on leaves and stems, prevents dehydration, while animals like birds and mammals have waxy secretions e.g., earwax to protect against water ingress. Previous year solved question. Which type of lipid is primarily responsible for forming the structural components of the cell membrane? A. Triglycerides. B. Steroids. C. Phospholipids. D. Waxes. Answer. Correct option, C. Phospholipids. Explanation. Phospholipids are the major components of the cell membrane. Their amphipathic nature allows them to form the phospholipid bilayer, which is crucial for the membrane's function as a selective barrier. Multiple choice questions. MCQs. 1. Which of the following is an example of a steroid? A. Cholesterol. B. Phosphatidylcholine. C. Triglyceride. D. Glucose. Answer. A. Cholesterol. 2. Which class of lipids is primarily used for long-term energy storage in animals? A. Phospholipids. B. Waxes. C. Triglycerides. D. Steroids. Answer. C. Triglycerides. 3. What is the main structural feature that makes phospholipids amphipathic? A. Presence of cholesterol. B. Presence of both hydrophilic heads and hydrophobic tails. C. Presence of fatty acids only. D. Presence of glycerol backbone. Answer. B. Presence of both hydrophilic heads and hydrophobic tails. This completes the lecture on lipids, composition, structure, and function. Let me know the next topic. You said, amino acids. ChatGPT said. Amino acids, composition, structure, and function. Introduction. Amino acids are the building blocks of proteins, which are essential for nearly all biological processes in living organisms. Each amino acid consists of a central carbon atom bonded to an amino group NH, a carboxyl group COOH, a hydrogen atom, and a unique side chain R group that distinguishes one amino acid from another. There are 20 standard amino acids that form proteins, and these amino acids differ in the structure of their R groups, which determine their chemical properties and function. Amino acids play critical roles not only in protein synthesis but also in various metabolic processes, enzyme activity, and signaling. The unique combination and sequence of amino acids determine the structure and function of proteins. 1. Composition and structure of amino acids. A. General structure of amino acids. Amino acids share a common structure, which includes amino group NH, a nitrogen atom attached to 2 hydrogen atoms. It is basic in nature and can accept a proton H in a biological environment. Carboxyl group COOH, a carbonyl group C equals O attached to a hydroxyl group O. It is acidic and can donate a proton. Hydrogen atom H attached to the central carbon atom. Side chain R group, a variable group that distinguishes one amino acid from another. The nature of the R group determines the chemical characteristics of the amino acid, e.g., hydrophobic, hydrophilic, acidic, basic. B. Classification of amino acids based on their side chains. Amino acids can be classified according to the nature of their side chains. Nonpolar hydrophobic amino acids. These amino acids have side chains that do not interact well with water, such as glycine, alanine, and valine. Polar hydrophilic amino acids. These amino acids have side chains that can form hydrogen bonds with water, such as serine, threonine, and glutamine. Acidic amino acids. These amino acids have a carboxyl group in their side chain, which can release a proton H, making them negatively charged at physiological pH, such as aspartic acid and glutamic acid. Basic amino acids. These amino acids have an amino group in their side chain, which can accept a proton, making them positively charged at physiological pH, such as lysine, arginine, and histidine. Aromatic amino acids. These amino acids have an aromatic ring in their side chain, such as phenylalanine, tyrosine, and tryptophan. C. Essential versus non-essential amino acids. Amino acids can also be categorized based on whether they need to be obtained from the diet or can be synthesized by the body. Essential amino acids. These amino acids cannot be synthesized by the human body and must be obtained from a diet. Examples include leucine, tryptophan, and methionine. Non-essential amino acids. These amino acids can be synthesized by the human body. Examples include glutamine, alanine, and asparagine. 2. Functions of amino acids. Amino acids perform a wide range of essential functions in the body, particularly in the synthesis and regulation of proteins, enzymes, hormones, and neurotransmitters. A. Protein synthesis. Amino acids are the basic building blocks of proteins. During translation, the sequence of amino acids determines the structure and function of the resulting protein. Each amino acid is linked to the next by peptide bonds to form a polypeptide chain, which then folds into a three-dimensional shape based on its amino acid sequence. B. Enzyme catalysis. Amino acids are crucial for the functioning of enzymes. The active site of an enzyme is typically composed of specific amino acids that participate in the catalytic process, either by stabilizing the transition state or directly participating in the chemical reaction. C. Neurotransmitter synthesis. Some amino acids function as precursors for neurotransmitters, which are chemical messengers in the nervous system. For example, tryptophan is a precursor for the neurotransmitter serotonin. Tyrosine is a precursor for dopamine, norepinephrine, and epinephrine. Glutamate is an excitatory neurotransmitter. D. Hormone production. Amino acids are involved in the synthesis of hormones, both peptide and non-peptide hormones. For instance, tyrosine is involved in the production of thyroid hormones thyroxine and catecholamines dopamine, norepinephrine, and epinephrine. Phenylalanine is a precursor for tyrosine, which in turn is involved in hormone synthesis. Acid-base balance. Amino acids can act as buffers in the body to maintain pH balance. The amino group can accept a proton in acidic conditions, and the carboxyl group can donate a proton in basic conditions, helping to stabilize the body's pH. Metabolism and energy production. Amino acids can be broken down to generate energy. When carbohydrates and lipids are in short supply, amino acids can undergo deamination, removing the amino group, and the remaining carbon skeleton can be used to produce glucose via gluconeogenesis or enter the citric acid cycle to produce ATP. 3. Peptide bond formation and protein structure. The amino acids in a polypeptide are joined by peptide bonds, which are covalent bonds formed between the carboxyl group of one amino acid and the amino group of another. The reaction releases a molecule of water, condensation reaction. The sequence and structure of amino acids in a polypeptide chain define the overall protein structure, which can be categorized into 4 levels. Primary structure, the linear sequence of amino acids in a polypeptide chain. Secondary structure, the folding of the polypeptide chain into regular structures like alpha helices or beta pleated sheets, stabilized by hydrogen bonds. Tertiary structure, the three-dimensional shape of the protein, formed by interactions among the side chains of the amino acids. Quaternary structure, the assembly of multiple polypeptide chains into a functional protein complex. Previous year solved question. Question. Which of the following amino acids is essential and cannot be synthesized by the human body? A. Alanine. B. Glutamine. C. Tryptophan. D. Asparagine. Answer. Correct option, C. Tryptophan. Explanation. Tryptophan is an essential amino acid, meaning it must be obtained from the diet as the human body cannot synthesize it. The other amino acids listed are non-essential and can be synthesized by the body. Multiple choice questions. MCQs 1. Which of the following amino acids has an aromatic side chain? A. Glutamine. B. Phenylalanine. C. Lysine. D. Glycine. Answer. B. Phenylalanine. 2. Which part of an amino acid gives it its unique properties? A. Amino group. B. Hydrogen atom. C. Carboxyl group. D. R group. Answer. D. R group. The primary structure of a protein refers to a. The folding of the polypeptide chain into alpha helices and beta sheets. B. The sequence of amino acids in a polypeptide chain. C. The three-dimensional shape of the protein. D. The interactions between multiple polypeptide chains. Answer. B. The sequence of amino acids in a polypeptide chain. This completes the lecture on amino acids, composition, structure, and function. Let me know the next topic. You said, proteins, ChatGPT said. Proteins, composition, structure, and function introduction. Proteins are fundamental macromolecules that perform a wide variety of essential biological functions. They are composed of long chains of amino acids, and their sequence determines their unique three-dimensional structure, which in turn determines their function in biological processes. Proteins are crucial for the structure, function, and regulation of cells, tissues, and organs in living organisms. They are involved in catalyzing biochemical reactions, enzymes providing structural support, such as in muscle and skin, transporting molecules, and regulating cellular activities. 1. Composition and structure of proteins. A. Amino acids and peptide bonds. Proteins are made up of linear sequences of amino acids, which are linked together by peptide bonds. A peptide bond is a covalent bond formed between the carboxyl group of one amino acid and the amino group of another amino acid, releasing a molecule of water. The resulting chain of amino acids is called a polypeptide. Each protein contains a specific sequence of amino acids, which is encoded by the organism's DNA. The sequence of amino acids in a polypeptide chain is referred to as the primary structure of the protein. B. Levels of protein structure. The overall structure of a protein is defined at 4 distinct levels, which are essential for its function. Primary structure. This is the linear sequence of amino acids in the polypeptide chain. The primary structure is determined by the genetic code and DNA and is the most fundamental level of protein structure. Secondary structure. The polypeptide chain folds into regular structures, such as alpha helices and beta pleated sheets, which are stabilized by hydrogen bonds. This structure is not yet the final form but represents the localized folding of the chain. Tertiary structure. The overall three-dimensional shape of the protein, formed by the interactions between the side chains, R groups, of the amino acids. These interactions include hydrogen bonding, ionic bonds, hydrophobic interactions, and disulfide bridges, covalent bonds between cysteine residues. The tertiary structure determines the protein's specific function. Quaternary structure. Some proteins consist of more than one polypeptide chain, each with its own tertiary structure. The quaternary structure refers to the arrangement and interaction of these multiple polypeptide chains to form a functional protein complex. Examples include hemoglobin, composed of 4 polypeptide subunits, and DNA polymerase. C. Protein conformation. The three-dimensional shape of a protein is often referred to as its conformation. The protein's conformation is crucial for its biological function because it determines how the protein interacts with other molecules, such as substrates, ligands, or other proteins. A protein's conformation can be altered by various factors, including pH, temperature, and chemical modifications. In some cases, denaturation can occur, where the protein unfolds and loses its functional conformation, often due to extreme conditions. 2. Functions of proteins. A. Enzymatic activity. Enzymes are proteins that catalyze biochemical reactions by lowering the activation energy required for the reaction to proceed. This is one of the most important roles of proteins in living organisms. Enzymes are highly specific to their substrates and operate through mechanisms such as substrate binding and transition state stabilization. Enzymes facilitate nearly all chemical reactions in cells, including metabolism, DNA replication, and protein synthesis. Examples include amylase, lipase, and DNA polymerase. B. Structural support. Proteins play a crucial role in providing structural support to cells and tissues. They are the main components of the cytoskeleton, a network of filaments that provide shape and mechanical support to cells. Structural proteins such as collagen, keratin, and elastin give tissues like skin, bones, and muscles their strength and elasticity. Collagen is the most abundant protein in the human body and provides structural support in connective tissues, skin, and bone. Keratin is found in hair, nails, and skin, providing strength and protection. Actin and tubulin are structural proteins in the cytoskeleton that help in maintaining cell shape and facilitating cell division. C. Transport and storage. Proteins are involved in the transport of molecules across biological membranes and in the storage of essential substances. Transport proteins such as hemoglobin carry oxygen in the bloodstream, while myoglobin stores oxygen in muscles. Ion channels and carrier proteins facilitate the movement of ions and molecules across cellular membranes, maintaining cellular homeostasis. Hemoglobin transports oxygen from the lungs to tissues and carbon dioxide from tissues to the lungs. Ferritin is a storage protein that stores iron in a non-toxic form, releasing it when needed by the body. D. Immune response. Proteins are integral to the immune system, particularly antibodies, which are proteins that recognize and neutralize foreign invaders such as bacteria, viruses, and toxins. Antibodies, or immunoglobulins, are highly specific and can bind to antigens, foreign molecules, marking them for destruction or neutralization by other immune system components. Additionally, proteins such as cytokines mediate communication between immune cells and help regulate immune responses. E. Hormonal regulation. Proteins also act as hormones, which are signaling molecules that regulate physiological processes in the body. Hormones like insulin, growth hormone, and glucagon are proteins that regulate metabolism, growth, and other vital functions. Insulin is a peptide hormone that regulates blood sugar levels by promoting glucose uptake into cells. Growth hormone stimulates growth and cell reproduction. F. Cell signaling. Proteins are central to signal transduction pathways, where they transmit signals from the outside of the cell to its interior. Receptors on the cell surface bind to ligands such as hormones or neurotransmitters, and this interaction triggers a cascade of intracellular events that regulate cellular responses. Examples include G-protein coupled receptors GPCRs, which are involved in a variety of cellular processes, including vision, smell, and immune responses. 3. Protein denaturation and folding. Proteins can lose their functional conformation in a process known as denaturation, caused by changes in environmental conditions such as extreme heat, changes in pH, or high salt concentrations. Denatured proteins lose their three-dimensional structure and become non-functional. For example, when an egg is boiled, the proteins in egg white denature and turn from clear to opaque. Protein folding is the process by which a newly synthesized polypeptide chain adopts its functional three-dimensional structure. This folding is highly regulated and can be assisted by chaperone proteins, which help prevent incorrect folding or aggregation. Previous year solved question. Which of the following is a function of proteins? A. Storage of genetic information. B. Catalyzing biochemical reactions. C. Transport of glucose. D. Synthesis of RNA. Answer. Correct option, B. Catalyzing biochemical reactions. Explanation. Proteins are responsible for catalyzing most biochemical reactions in the body, acting as enzymes. While DNA stores genetic information and RNA helps in its expression, proteins play a direct role in catalyzing reactions, transport, and other cellular functions. Multiple choice questions. MCQs. 1. What determines the function of a protein? A. The sequence of nucleotides. B. The sequence of amino acids. C. The sequence of fatty acids. D. The sequence of sugars. Answer. B. The sequence of amino acids. 2. Which of the following is not a function of proteins? A. Structural support. B. Energy storage. C. Transport. D. Enzyme catalysis. Answer. B. Energy storage. 3. Which of the following is an example of a protein involved in immune defense? A. Hemoglobin. B. Antibodies. C. Ferritin. D. Insulin. Answer. B. Antibodies. This concludes the lecture on proteins, composition, structure, and function.